right, this is the student view of the lab, of the Connect lab portion. Hopefully you can, when you log in to Connect, you'll find something, you'll either see this or you'll see something that lets you click on assignments and come to this page. I do not know what you see on the first page that you get, but if you see something called assignments, click on it and hopefully it brings you here. I do not have a student view of what you have on your dashboard. It may just be this. If it is, great, and you're in the right place. Now, you'll notice that you have Lab 1, 2, 3, and 4. This should be what you see. Please send me an email if you do not see this in your assignments page. Let's see, what else do you need to know? Notice when the dates here, basically I'm going to give you a week to complete each of these. They're not that bad. September 8th to September 14th. All right, so that's 7 days for you to complete this one. Lab 2 starts on the next day, so Thursday is the 8th. You have a week to the 14th to do this, and you'll also have 3 attempts to do it. So you can do it 3 times, and hopefully you do better on the last one because I think it'll accept the last one. So September 9th to the 15th, so you have a week there. So you have 1 week to complete these 2 essentially. You get 1 day later, so you can do it this one, and maybe you complete it on the 10th or 11th, and here you start this one then, and you have all the way to the 15th. Or you can just do them both all the way, any way you want. This one is due the 14th, and this one is due the 15th. This one you have access on the 8th, and here you have it on the 9th. Same way next week, I split it by a few more days, the 12th and the 14th, due on the 18th and the 20th. By the time we get here to September 20th, we will be ready for an exam. So now these last 2 are just crosses anyhow, so it'll just be going back to review you on stuff you've already done. We'll have moved on to other material, but it's always good to review these crosses right before you take the exam. So I think this is a good idea. Now, it's locked now because this doesn't open to the 8th, and I'm making this on the 7th. So we cannot get in, but I have already started it over here, so you can see when you click on the arrow that will be right here, you will go to Lab 1, and you should see this. So here we have information, and you're going to be doing this lab simulation. This will be your laboratory, and it will grade it automatically. I won't have any feedback on it, but I won't have any control on how the controls work. You have to figure out the controls, but I'm going to try and help you out a little bit here because it can be confusing. Now here you've got key concepts, overviews, make sure you're aware of what all of these terms mean. You will need to know this stuff for the test. So this is good stuff to kind of write and take notes on. In fact, you probably should kind of be familiar yourselves with what is this: prophase, metaphase, anaphase, telophase, interkinesis. I'll tell you right now, the thing to remember is PMATI, PMATI, and that'll help you as you move on through these, at least through this simulation. And now this one down here is PMAT and daughter cells. So just realize PMAT, P-M-A-T, are the ways, the way you remember the order of meiosis. Now, you'll need to understand it in a little more detail for the test. I do not focus a lot on this, but you do have to at least know the order of the stages and generally what they look like. Now, here are some definitions, by the way. The ones that are useful for you are homologous chromosomes. I'll use that quite a bit. Sister chromatids sometimes. Autosomes, I will use diploid and haploid. You need to know those for sure. So the last 2 have to do with these stages. Probably won't ask you a whole lot. But these first 4 terms you should probably know. Now when you're ready, you kind of reviewed this a little, maybe gone over the book and tried to figure it out, maybe watch a video on YouTube, something. You need to go and you'll come to here. And this is your lab, your simulation. So you can, on a PC, use Control-Alt-K to skip ahead. But I'm gonna do the first one here for you. So what are we trying to do here? Well, we're going to do normal meiosis, and you're going to do the rest of them your own by yourself, but I'll do this one with you. So what did I just tell you? What were the things? PMAT, PMAT, I. So how do we put these? We need to put these in the right order, and the way you do that is prophase. Oops, it gives you an error. So click once, and then it moves. Don't double click, or don't drag either. You click, let go, and then drag. And then I'm going to drop it here. P, and then it's mat. Click. M-A. T. And then I is last. So we got that. Now, we also have to put the cells in the right order. So you click, let's see, we'll guess here, and which one do you think goes first? Well, I'm not sure which one goes first, but I'm pretty sure the last one has the two final cells. And I think right before that, these have come together and they're here. Now I'm just going to randomly put the others in here. Oops. And we'll see, they're probably wrong. So I can show you. I know these are right, but let's see. So we've got two wrong. All right, so what does that mean? Well, we have to change them. And this is going to be easy because obviously if this one is here and this one is here, the other one is in the other spot. So what do? Well, close this box. Put this one over here. Put that one over there. And we got it right. Now, when I first did this a couple hours ago, I kept trying to click over here and go to the next one. There's a question you have to ask, or you have to answer. So what is the right answer for this one? Well, if it's a square box, it means there can be multiple correct answers. I think I know the right ones. Let's see if I got that right. Partially correct. No, I've forgotten which numbers they were. These cells are diploid. Which describes the cells at the end of meiosis 1? And they are homologous. Incorrect. So you're going to have to, like I said, let's just go through it. Partially correct. So why don't we do this one? There we go. So through two chromatids connect to the centromere, the cell, okay, that's describing haploid. And the second one is two homologous chromosomes of homologous pair have separated and moved to create two newly cells. Yes. All right, so then you can go, after you get these correct, you can go correct. And now you can move on to the next part. All right, which would be, maybe normal meiosis 2. And now you have to put these pictures in the right order. I don't know what they are at this point. I'm just moving them so we can get some more practice. And we'll just put everything, I'll mix it up a little. We know what the right answer is. I know that one is down here. No? What is it? Is it wrong? Actually, yes. That one's right. The rest of them, no, these are right. I just get it without even paying attention. I got most of them right. These are wrong. What is it? PMAT, and then whatever the last thing is. So, of course, this one should go over here. This one should go here. This one should go here. Go here. And if these were wrong, then we switch these. Just to remind you how it works. That should be correct. There we go. And now we have another question. We have, what, four haploid cells. 4 haploid cells? Correct. There we go. We can continue. All right, and that's how you do these labs. So you continue on, learn these phases. This is stuff, I don't know, I learned it in high school. I don't know when you might have seen this, or maybe you haven't, but you'll need to complete this lab. And as we get closer to the exam, I'll tell you what parts you need to know. I can tell you right now, I would know P, M, A, T, and then something else. All right, something else will go at the end usually, but PMAT. Memorize that right now. And that's your lab, that's your first lab, and all the labs will be similar to this in some way.